Morning, everyone. Welcome to the H2NOW Chicago Town Hall, where we're really excited to share the latest in real-time water quality monitoring for the Chicago River and take your questions about this new tool and how it can help you make better choices about how to interact with the Chicago River. My name is Elena Harkness. I'm the Executive Director of Current, and I'm here today with the team from Current to walk through our H2NOW Chicago website and share a little bit more about how we built it, where it is today, how you can interact with it, and then take your questions to help guide our further development of the tool and also just help you understand how to use it better. So before we kick it off, we've got a poll that's launched, and we hope you'll share with us what's your current perception of the Chicago River. You've got some options here, so please go ahead and take a moment to fill that out. All right, good participation so far, that's great. Your options are very poor water quality, fair water quality, good water quality, and very good water quality. So take your best shot. Thanks, folks. All right. Thanks to everyone who participated. Sounds like a vast majority of the folks in the room today think that it's fair water quality. So that's subjective. There's a lot of room to talk about what that actually means. But the whole point and purpose of H2NOW Chicago is actually to share with you what we're learning about what's really going on in the river. So without further ado, oh, great, George, thanks for sharing the poll questions. Perfect. Just a little bit of background about Current and why we're here to talk about real-time water quality monitoring today. Current's a nonprofit water innovation hub headquartered here in Chicago, Illinois, and we're about 5 years old this year, and our job is to drive collaborative innovation to help grow the inclusive blue economy, accelerate innovative water technologies, and solve persistent water problems. And one of those water problems is the water quality of our urban rivers. So H2NOW Chicago, the QR code on your screen will take you to a website that opens up a portal that I'm going to show you in just a moment. And H2NOW Chicago, we like to talk about it like a weather app for the Chicago River. It's going to show you in real time an estimate of fecal coliform levels in the river. And we like to call H2NOW Chicago a partnership powered platform because it is really dependent on many different partners to make it work. And its goal is to advance real-time water quality monitoring and communicate that data about the river's health with you, the public, and our partners that help make this work. Here's a preview of what the gauges look like. We've just rolled these out to the public and we're really excited to go into more detail. But as you can see, it's a snapshot that shows you at any given time what's going on across the three main branches of the Chicago River. On the left, we have our main stem. And for those of you who are not familiar with the Chicago River, the main stem cuts right through the heart of the loop. And it's the place where our Chicago River Walk has been really built and improved in the last decade or so. And it's the part of the river that most Chicagoans are probably most familiar with. We also have the North Branch, which runs all the way northwest into the northwest corridor of the city, and the South Branch, which runs south and west. And so H2NOW Chicago, one of our central goals was to understand how water quality is changing across all three branches of the Chicago River because it does cut through all of our city. As I said, H2NOW Chicago is only possible because of the contributions of many different partners. Here you'll see our core public utilities, which have been really the anchor partners in bringing H2NOW Chicago to life, the Metropolitan Water Reclamation District of Greater Chicago, the City of Chicago's Department of Water Management, so many of our region's research universities, technology partners, advocates that have been really driving forward this vision for a great and productive river system here in the city of Chicago. And of course, our core funder for this project, the Chicago Community Trust, its Great Rivers Chicago program. So all of these partners were so grateful to have helped us bring this project to life. Just a bit of the timeline, because while we're excited to rush right into the today status and show you the gauges, just want to give you a sense that this project took a long time to bring to life. So in 2015, the Metropolitan Planning Council and the Chicago Community Trust launched the Great Rivers Vision for the region. And you can go check this out. It articulated many different goals for our regional river system, including the Chicago River. And it had goals for 2020 as well as a vision for 2030. And all of them are aligned around this vision of living, productive, vital river systems here in our region. One of those goals was real-time water quality monitoring, and Current, as an innovation hub in Chicago, picked that as one that we thought we could help bring to life. So between 2018 and 2021, we worked on acquiring the technology, setting up the platform, testing it to understand if it would perform in this sort of real world setting of the Chicago River, and then developing the front-face tool that helps us communicate all of this data to the public. And then we were very excited right before Labor Day this year to finally be able to launch to the public. So I'm going to go ahead now and switch over to our H2NOW Chicago website. You can pull this up on your mobile phone. You can pull it up on your computer yourself. This is what the website looks like. It's going to give you a homepage view that takes you right to the gauges. So here you go. This is the latest water conditions as of 10:52 this morning. So, really within the last just about 15 minutes here, and they're probably about to update again. What these gauges are showing you is a range of water quality estimates, and our gauges are based on sensors that really tell us a combination of three things. They're tracking temperature, they're tracking turbidity, and they're tracking something called tryptophan-like fluorescence. Tryptophan-like fluorescence is a compound that is found in all warm-blooded organisms. It's the same thing that makes you tired from Thanksgiving turkey. You might have heard of like tryptophan in that context. It's the thing that makes us all take naps after we've had our Thanksgiving meals. But tryptophan is something that, because we find it in warm-blooded organisms, it is being tested as an estimate of something else that warm-blooded organisms produce, fecal matter. So fecal matter in the river is something that we care a lot about, not necessarily because it's itself something to be concerned about. Fecal matter is actually found in all river systems. It comes from warm-blooded animals. It washes off into the river from, birds and animals and other, creatures that are making their home in the river. But it also comes from humans, and it comes from sewage. And so when you sign up to receive a combined sewer overflow warning from the Metropolitan Water Reclamation District or our friends and partners at Friends of the Chicago River, that's an indicator that there's been an overwhelming event that has caused our combined sewers to empty into the Chicago River. So these are massive events that don't happen very often. They do happen when we have really extreme rain events in the region. And so one of the things we are watching for and need to care about is more and more extreme rain events that are coming with climate change's impact on our region. We need to think about how that's affecting our river systems and how it's affecting our infrastructure. So again, as I said, you can sign up for those alerts. You can find out when those extreme events have happened. What our gauges are telling us is what's happening below those extremes, right? So how the river is fluctuating across the branches in real time on a daily basis. And what you can see here, if you take a look at the main stem, the north branch and the south branch, is it's really different. It looks different across the branches. And the labels that we have, good, low caution, and high caution, are there to give you a little bit of a sense of what are the kinds of activities on any given day that you might want to be doing in the river. So on a low caution day, that means that the water conditions we're observing might be slightly higher than the recreational contact standard. That's something that the EPA sets that says basically if the water conditions are meeting or exceeding the recreational contact standard, that's an okay day on a primary contact standard day to actually swim or bathe or surf. That's what primary contact standards mean. So we've chosen to use the EPA's primary contact standard as our indication that the water quality is good, meaning it might be, safe to actually swim in. But on a low caution day, that means it's might be above the primary contact standard, but it's still in a range that we would consider, low caution to do things like kayak, really like walk along the river, enjoy a day of, eating alongside the river on the river walk. Low caution is really when we've been looking at the trends over time, mostly this is what the river's looking like on any given day. It's kind of in this low caution range. So not a day you'd want to be swimming, but find a kayak. Right now, looking at the South Branch, that's in a high caution moment. And that means it's above the recreational contact standard, but it's also sort of above where we'd want to see for kayaking and that kind of thing. you can still go out on the river, but we want you to know that this is a day where the parts per billion of tryptophan-like fluorescence is higher than we might normally see. And so these gauges are really designed to give you a sense of the average, so what we're seeing in a branch on a monthly basis. That's important because these branches are highly variable, and what we care about is kind of how the quality is changing relative to how it might be on a normal day. And you see that the averages vary quite a bit, main stem, north branch to south branch. There's a lot of reasons that that's the case. River systems are really complex, and if you think about the volume of water that's flowing through the main stem, a lot of that water is lake water. So when you see the low caution at only 5 parts per billion, and then low caution for the north branch is still much higher, that's because on an average day, the background conditions in the north branch are very different from the main stem, and they're very different from the south branch. So I'm actually looking at Kelly's question in the chat. Why is north branch low caution when the values are higher than the south branch, which is listed as high caution? That's the reason for that. The background levels are just different as a sort of regular, daily background limits in the north branch and the south branch. And so what we actually would read as a measure of high caution, you can't take those numerical values. It's not like, 5 is good and 50 is bad. It matters a lot what those background conditions are. So please keep your questions coming in the chat. These gauges are designed to be kind of the front door to H2NOW Chicago, and the, really at a glance way to say, good day, maybe not such a great day to kayak. What's it looking like over time? But I'm now going to turn it over to our technical program director, Svetlana Taylor, and I'm going to go a lot deeper. We're going to go over to the data page where you're going to be able to understand even more about the data, and she's going to explain a lot more about how we got to these gauges and how we arrived at the data that you see today. So I'm going to zoom in on the real-time data, and I'm going to turn it over to Svetlana. Svetlana, over to you.
Go to the data page.
Yep, hold on one second. I'm navigating. Here we go. Great. Okay, so on our data page, we have detailed branch readings for each branch of the Chicago River. Here we go. Svetlana, do you want to walk through what the probes measure and what the data means really quickly?
Yes, I'll do that. Hello, everybody. Thank you for being here with us today. I will explain, I'll try to explain what our sensors actually mean, what we can see, and how to understand it. So this first plot is the tryptophan sensor readings. And as Elena mentioned, we're measuring tryptophan-like fluorescence, or we can just call it tryptophan, which is an amino acid that fluoresces, and that's how we measure its concentration. Mm-hmm. So tryptophan is a parameter that responds to microbial concentrations. But it's not a direct measure of fecal coliforms. So Elena mentioned fecal coliforms is what the standards are. We're measuring an indirect parameter that we think corresponds to fecal coliform, but it's not directly corresponds. So we're still really trying to understand how it corresponds. And our assessments, high caution, low caution, are really based just on the fecal coliform data that we've been able to collect over the summer. But there's still really rough estimates right now because we're trying to be very conservative. And if we saw a high fecal coliform level, we use the lowest tryptophan level that we observe as our guide. So sometimes, the water quality may actually be better than what we are saying. And really what we're trying to go from, what we are more confident in is how the tryptophan responds. We can see those peaks. That means something is happening, and there's probably more microbes in the water at that point.
I'm pointing out a peak. Sorry, there's a peak right there. This is a tryptophan peak that happened in August. Yep.
Yeah.
Go ahead.
If we can move to plot 1, actually, that would be great.
You bet.
Plot 1. So on plot 1, where we have data explanation, plot 1.
Oh, sorry.
Lower than that. Yeah, that's the one.
Here we go. Sorry.
Perfect.
Great.
Thank you. Yeah, so we can see that during dry weather periods, tryptophan levels usually stay stable, or they may trend upwards or downwards. Sometimes it just depends on the water temperature, and if the weather is getting warmer, we may see just more microbes in the water. But when we have rain events, almost always we observe a peak in tryptophan, and it usually happens about 12 hours after the rain. So something happens in the water that makes kind of maybe a delayed reaction. But these are the times when most likely the microbial concentration in the water is getting higher. And at the very end of this plot, you can see a very steep rise in tryptophan, and this is what we think is a response to combined sewer overflow. And the reason I'm saying it because we've seen that in the previous years. We've tested in 2019 and 2020, and we've seen a very similar rise every time a CSO was reported upstream of our probe. So we think it's very likely that that was a response to CSO. We can go to the next plot, to plot
Great.
2.
And just 1 second before rolling off this plot. So I think one thing that's important to note here, this CSO event, when these happen, again, these are what, with investments in infrastructure and other, other things in the region, these are what over a period of many years, these have been declining. So these used to happen with more frequency, and they've been declining, which is great. These are the alerts that you can get on your phone when they still do happen periodically. But it is important to look, this is what we're interested in, is these other spikes that might not be from a CSO, but are from other extreme weather events, and how those might affect your choices to, use the river on a given day. So it's, you can see there's a lot more granular data behind, these major CSO events, which we hope, we get to a point where those never happen. But in the meantime, it's really important to check what's going on beneath that in greater levels of granularity. So great, thanks for that, Svelana. So where did you want me to go next?
Yes, next plot, that's where we actually included our fecal coliform data for that time period. So you can see that when those peaks occur after rain events, we usually see higher concentrations of fecal coliform. That blue line on the bottom, that's the line that corresponds to 200 CFUs per 100 milliliters of fecal coliform. That's the measure of fecal coliform. And if we see that is above the line, so it's above this standard, and most of the readings during that period that we took, they were above that line. You can also, if you can see at the end of this plot, after that suspected CSO event, readings are very high still. You can see the fecal coliform even 2 days later is still very high. And so even though the tryptophan may be lower, we decided, because we really are not sure what is going on, but we see this high reading, so we decided that we'll keep high caution label for 3 days, even when we suspect that CSO has occurred or a CSO has been reported. So you may see lower tryptophan levels on the gauge, but still a high caution reading. And on plot 3, we kind of can zero in to see what's actually going on with other water quality parameters during a CSO event. And what we always see is that turbidity also rises. You can see a really high peak in turbidity here that actually occurred slightly before the tryptophan peak, and the temperature also rises significantly with these events.
Hey, Svetlana, everybody I think knows what temperature is, and you've explained tryptophan, but could you explain just turbidity for folks who might not have a sense for what that means in terms of a measure of water quality and why we care about it?
Yes, of course. The way I like to explain turbidity is a measure of murkiness of water. So if actually in the past, the way turbidity was measured, there was a circle that was lowered in the water, white circle, and at some point you will not be able to see it. So that's what turbidity is. If there's a lot of solids, dirt in the water that mixed in, then we have high turbidity.
Great, thanks.
Of course. Okay, we can go to plot 4.
Okay.
So sometimes on the North Branch, water from Lake Michigan enters the North Shore Channel. And when that happens, we can actually see a drop in tryptophan. We also see a drop in temperature. And we've observed that actually twice, once during this time in May, and then we also saw that just happened recently. So that's something that we don't know exactly what's going on with fecal coliform during these events, but it's interesting that we can observe that in our sensors. And we can go to the next plot. So this plot is our depth plot. We have depth measurement, so we can kind of see what is going on with the levels in the river. Obviously, when it rains, the levels will get higher. What we also see, we know that Metropolitan Water Reclamation District controls the level of the river. They watch the weather forecast, and when they know that a lot of rain is expected, they'll lower the level of the river. So we often observe that happen before rain event, and then the level will rise after a rain event. And I don't know if you remember, on June 26th, we actually had a really large storm event, and actually I have never seen the level of the river rise that much, the one that you see circled here, but just thought we pointed out that that's something we also can see.
Excellent.
That's all. I think we've gone through all the plots on our data explanation page, and if you have any questions, I'll be happy to answer them.
Great, thanks so much for that, Svetlana. And you can see we've really built this kind of 1.0 iteration of the H2NOW website to allow you to explore the data and to go a little bit deeper, right? So you can check and see what's going on in the main stem, what's going on in the South Branch, the North Branch, but these explainers that we have are designed to help walk you through what is actually some very complicated science. So doing river quality monitoring is, in real time especially, is really hard. And one of the things that we want to make sure everybody understands about this is that it is a novel pilot project. So we talk about H2NOW Chicago as a platform for testing water quality monitoring technologies, and that's because everything that is involved in this process, I'm actually going to walk you just through the pieces that are involved. They're all pretty novel, and we're testing how they perform in real time. So we're doing this publicly with all of you because we think there's so much rich information to learn about the river, and it's frankly so much more than has ever been available before, which is really, really exciting. But we're also asking you to bear with us, as there's going to be moments where different pieces of this technology, might be experiencing an outage, as we had over the weekend, where some of the telemetry wasn't updating as much as we needed it to, or there's other reasons that we might have to pull down the data from time to time. So that's something to be expected. It's real-time science that's happening out in our backyard in the Chicago River. But here's just really quickly some of the different pieces that are involved and at stake that we're out there testing. So the sensors themselves across the bottom here, these are the sensors that contain, well, they're called probes, and they contain the different sensors that Svetlana just walked you through that track temperature, turbidity, and the optical sensor that is estimating this tryptophan-like fluorescence. So the optical sensor is the most novel of those pieces of technology. And it's novel not only because of the sensor, but because we're actually working with this company to try to understand how well it estimates fecal coliform. So that itself is something that we're testing in real time. So these probes sit in the river at 3 different spots, as I said, main branch, south branch, north branch, and then we have to actually receive the data. So all of these different components, RS Hydro is the company that works to make the Proteus probes. They have some telemetry that actually communicates what the sensors are reading. IECA is another platform that's helping us. It has a wavelet technology that helps us communicate the data to a data visualization platform. And that is called the iGreen platform. Our partners at IOSite have made that for us. And it's what helps us take the raw data and convert it to something meaningful. Then finally, we do the gauge design and we have everything presented in a way that actually is actionable to the public on the H2NOW Chicago front-facing website. So there's a lot of different pieces, even in this kind of 1.0 phase that we've rolled out now, and we'll be optimizing for that as we move forward into the future. So that's a little bit on the technology and how this is all rolling out. I am going to pause because we've got some great questions coming in from the audience, and I'm just going to actually go back to our data page to keep the gauges up in the background while we do that. Let's see. Here we go. Back to our gauges. Okay, excellent. And yes, see, they've updated even since we've been on this call, on this webinar altogether, and now we've got water conditions as of 11/22. Great. So we've got a question from Justin at MWRD. Could we explain why we selected fecal coliform as the health indicator to calibrate for water quality versus E. coli, which is measured to indicate water quality in Lake Michigan and other freshwater systems? So I'm going to, oh, Svetlana's typing an answer, but I'm going to ask her to go ahead and answer that live. Svetlana, do you want to answer that for everyone? I think this is something that is really of interest. Everybody asks why we have picked fecal coliform, and at the very highest level, it's because they're a great indicator organism. And if there's more fecal coliform, we're interested in understanding what that might mean about other organisms in the river. But Svetlana, you want to jump in here?
Yes, definitely. I will answer this question. It's a good question. The reason we chose fecal coliform is because that is the indicator that was selected by IEPA for the Chicago River. So it wasn't our choice. There are several indicators that are used for assessing microbial water quality. So E. coli is what is used in the beaches, Justin pointed out. Also, streptococcus is another one that is often used, and there are many others that are being explored as good indicators. So basically trying to find an organism that's easy to measure and that can predict the level of pathogens being present in the water and associated strongly with human content. So fecal coliform actually is maybe considered not as good of an indicator, but we chose it because that is the measure that IEPA is currently using.
And that's the Illinois Environmental Protection Agency, for those of you who may not have known that or work with the IEPA as much as probably some on this webinar do. Thanks for that. That's great. And thanks for the great question, Justin. We have another really great question from Steve Close at True North Venture Partners. So can these data be used to determine what causes river water to be less healthy? And then can we use those insights to figure out what can be done to improve river health? So that is a great question. And I've got a couple of answers to that, and then I'd love to have the team chime in. But so number one, we already know a lot about what's going into the river and the sort of sources of river contamination. We know that runoff from urban streets is one of the big contaminators in urban rivers, and it's a problem not just in Chicago, but in cities around the world. And one of the solution sets for that is green infrastructure and thinking about how to put in more native plantings, things that absorb heavy rainfall rather than diverting it onto streets and impermeable surfaces like pavement, where it's going to pick up a lot of other contamination and dump it into the river, right? So if you think about having more absorbent capacity along our river edges, we've started to see a lot more of this along the river walk. That's really exciting. But having our edges of rivers be, less concrete and more green is one of the big solution sets that we already know is out there. So that's really important to talk about. But another thing we're really excited about with H2NOW is that because it's a platform for real-time water quality monitoring. We're testing other kinds of technologies that can help us answer with more granular detail, what are some of the sources of fecal coliform that are going into the river? How do we disambiguate between fecal coliform coming from, say, animals and fecal coliform coming from humans? And Svetlana, do you want to talk about our new pilot with Veriset, which is a technology that we've actually added to the H2NOW platform in the last year? And we're really kind of excited to share what we're learning from that pilot technology.
Yes, sure. So there are microbial or molecular methods of testing microbes. This is what is used, for example, for testing COVID. If you have PCR, and so this company actually tests the microbial samples on a genetic level, and they have a database of signals that correspond to specifically to animals or humans, and they can distinguish even between different animals. So we collected our samples, we partnered with UIC to process them and freeze them, then we sent them to California. They processed them when their labs opened after COVID, and then we just got the report, so we're still looking at the results, but this is interesting to see where these fecal coliforms are coming from. So we'll share those results when we have finally time to look at them, and we'll share the results.
Yep, and we've gotten all kinds of other interest from partners that want to understand more about things like dissolved oxygen levels in the river, which is a really good indicator of how healthy the river is for some of the aquatic ecosystems and the animals that rely on it for health. So there's room to develop, and we'll talk about sort of where we want to go with this in the future, but there's room to add on lots of other technologies. And the whole point of having this collaboration is because we want the public to understand the data and be able to use those insights to drive improvements. So it's a great question, and we're really excited to continue to add on not only technologies, but the kinds of partners that can help us turn that information into actionable recommendations to help continue this trajectory of urban river improvement that we've been seeing over the last decades in Chicago. So we have another great question from Jessica about how the probes actually sample the water of the river. So I want to share a little bit more about that. And you can, at any time, you can go on our technologies page and look at some of the major technologies that are responsible for bringing H2NOW to life. So the Proteus multi-probes that I talked about, that's these guys over here. We actually have those installed in the river during the recreational season. So that's basically March to November, say, while, that's the time when all of us are mostly, out using the river, and it's the time when it's got the highest concentration of users. So they are installed in the river, and they are constantly optically scanning what's passing by them. So they are picking up and transmitting to us these estimates of temperature, turbidity, and tryptophan-like fluorescence. That's what's coming into the data. So they are just out there. This picture over in the corner shows actually somebody pulling the probe out of the river. That's what it would look like if we were doing maintenance or installing or deinstalling for the season. But they're otherwise actually, they're installed, they're all season, and they are constantly talking to us. So that's how they do the transmission, and that's how they're in there. The other thing that you will see occasionally, and I'll flip back in a moment to our slides, but you'll see the current team out sampling the river. And so that's actually, we're out dangling our own little collection cup. We're getting a sample of the river. It's called a grab sample because we're out there grabbing it. We're actually taking water out of the river, and we're doing a more conventional test to understand what's in it. That's how we actually got to the point of saying we're confident enough in this novel technology, the Proteus probe, in what it's telling us. We're validating that with the water samples that we're pulling out of the Chicago River and testing ourselves. So that correlation is what was happening over the 2020 and 2021 seasons, where we were trying to understand, is this probe a good enough estimate for us to feel comfortable sharing the data that it's sharing with us with the public? And so that's a little bit about how we've, the current team and our partners with, MWRD and others, we've been validating and vetting what the Proteus probes, which are pretty novel technologies, have been telling us. So hopefully that answers your question, Jessica, about how they're testing the water, the probes. We're sampling, they're testing. Great. Other questions, please keep them coming. And I'm actually going to flip back over to our presentation just to remind you that here's where we are. We have launched this to the public. We're really excited about that. And please share the word, spread the word about the website, check it out. It looks really fantastic if you've got it on your mobile phone. So just keep it bookmarked and you can, keep checking in daily to see how the river's doing. It's especially interesting to see what's going on after, periods of very dry weather, periods of very wet weather, because, just like a weather report, the weather matters for the river. So what's next? I just want to give a little bit of preview about what's coming up, because we have, even though we've been working on this behind the scenes for several years now, we are excited that we finally got this up and launched to the public during a recreational season. And what's next? So on screen, you can actually see our team at various points with our masks during COVID out there taking those grab samples that I mentioned and going to test the water. But we're going to be leaving the probes up through the end of the recreational season. We'll pull them out for the winter. We're going to do some, maintenance and make sure that they're still performing and how we'd like them, performing how we want them to be performing. We're actually going to talk about what comes next and how we might want to add on some technologies to test during the 2022 recreational season. Because as we said, it's a real-time science and innovation project, and we want to keep adding on. So we've got lots of queries from partners already about other things they'd like to see. So we'll be building out that plan over the winter. And then we also are really excited to be soon launching an STEM curriculum that's modeled on H2NOW Chicago, because we think it's really exciting to learn what's in the river in real time. And we think that youth all over Chicago will also think it's really exciting. So I want to turn it over to our program builder, Kalindi Parikh, and she's going to walk us through a little bit of what we've got going with BlueEDU, which is our STEM curriculum that's modeled on H2NOW. So Kalindi, you want to take it away?
Sure thing. Hi, everyone. Thanks so much for joining us today. My name is Kalindi Parikh, and I'm the program builder here at Current. And I'm excited to share a new program with you today that uses H2NOW in a really new and exciting way. For a little bit of background, BlueEDU is a compilation of water and STEM focused online learning tools for students of all ages. So if you go to our website here, just on this, where you see this arrow right now, you'll kind of find that list. And we began compiling and sharing this information in early 2020 as a response to the sort of sudden transition to virtual learning that happened because of the COVID-19 pandemic. But if you go to this section of our website, you'll actually find great resources for all ages. They're kind of categorized by age and by partners. Many of our partners have kind of contributed to this list, and there's just a really great wealth of resources. But, however, in compiling this list, we noticed a little bit of a gap in resources specifically related to water quality in our region, and specifically for Chicago public schools. So if you can go to the next slide. So as a response to this kind of gap, we created a new program, which we called River Lab, which is a completely virtual curriculum for high schoolers at Chicago Public Schools. This program was developed with the help of a curriculum consultant and in partnership with an advisory council of CPS teachers. And it really took shape as an interactive virtual field trip of the Chicago River. So if you kind of see some of these images, students can actually choose their own adventures and kind of navigate the river. And learn about water quality, Chicago's water infrastructure, water stewardship, and even water careers. So you'll kind of see some of these slides where they can kind of choose their own actions. It's kind of not quite as good as being there in person, but of course, still quite a lot of fun to use. And they'll kind of learn about, if they click and go fishing, they'll learn about, some of the wildlife in the river, and they'll learn about the great, public infrastructure projects like the Riverwalk, as well as some other really exciting topics. If you can go to the next slide. So students who participate in this river get to use real H2NOW data and explore our platform and our gauges to answer questions about water quality and learn about their role in keeping our waterways clean. One of the assignments available through River Lab also lets students watch interviews with water scientists in Current's network and explore potential careers and educational paths in water. So you'll see 3 of Current's great partners and collaborators here. We've got about 20 of these videos where folks can share, what got them interested in their water career and what it took, and sharing advice for students who might be interested in pursuing that kind of career. So what we really hope is that this program will inspire interest really young in water careers and showing young people in Chicago clear pathways from that first, piquing their interest, to possible, educational pathways and employment in the blue economy. So this program is really just the first of hopefully many exciting opportunities to use H2Now as an educational tool. River Lab is free. It's a totally free resource. So we're launching it later this fall. So please feel free to spread the word once we do that, and stay tuned for our launch, and definitely get in touch if you'd like to get involved. But we are really excited to share this and hopefully bridge some gaps in STEM education in Chicago.
Thanks so much, Kalindi. Yeah, we are really excited about this, and we hope that many of you will spread the word among the young water lovers in your life, and give us feedback, because we've built this in partnership with many of our current partners and supporters, and we've had some great beta testing feedback, but we are definitely looking forward for more and to build this out to, again, as Kalindi said, eventually we really want this to be a world where every student in Chicago, every youth in Chicago has a chance to become inspired by our waterways from the earliest age, and stay engaged and connected and learn about all the different career pathways, whether they be STEM, our advocacy, community development. There's so many ways to think about building careers with water at the center. So really excited. Nicole had a question in the chat. BlueEDU, what have you done with respect to conservation? So I think just a background around BlueEDU, for us, it started as just a collection of other people's curriculum resources, because in the pandemic, we found there wasn't one place you could go if you were a parent or an educator or a caregiver who really just wanted to find ways to connect the youth that you work with to water. So there are some conservation focused modules on the BlueEDU site, if you go take a look at those, that were created by other partners. But in terms of conservation messaging, Kalindi, I don't know if you can speak to some of the different choose your own adventure deep dives, but I know there are some that talk about the role of conservation and stewardship in increasing the aquatic habitat health, and also along the adjacent river edges, and bringing back animals and all those sorts of ecosystem indicators that we care about. But I don't know if there's other specific conservation messages that you'd want to share.
Yeah, so one of the really exciting parts of this curriculum is that it really is hyper interactive in the fact that students get to choose to do deeper dives into whatever topic is interesting to them. So for example, if they do go fishing, there is a link to a partner organization that really, kind of hones in on that topic. And if students have that interest, they can go explore a little bit deeper about that. Or if they're really excited about, the MWRD and our infrastructure, they can watch the MWRD's virtual tour. So we really think of this particular River Lab curriculum as kind of, it points people out in the right direction to learn about those other subtopics, for sure.
Great, thanks. All right, well, we do still have a little bit more time for Q&A, so if you have questions, go ahead and put them in the chat. And yes, big shout out to the Northwestern students in the design thinking class who did come up with this entire idea for the Choose Your Own Adventure River Lab concept. And it is great because it's been so scalable. It's really sort of a way that we can continue to build out pages. And yeah, just loved working with our partners at Northwestern and the design thinking students that helped us with that. So thanks, Felana, for that shout out. Just to point to a couple of upcoming opportunities, and there are many, many others, but if you're interested in getting involved with H2Now as an individual, you can always help us spread the word. So hashtag H2Now Chicago, share out the website, tell your friends and family. We've had some really great coverage of H2Now in the launch phase, which has been wonderful. You can find all of that on the media page of our website, but the Tribune wrote a really lovely article about water quality monitoring and why this matters. Yes, go Wildcats. Thanks, Cheryl. And we've also just actually today had a chance to be on Reset just this morning with Sasha Ann Simons at WBEZ. So check out that piece. It was with our friends from the Metropolitan Planning Council, as well as the Forest Preserve District of Cook County, which is great. There's so many ways to be out there accessing our rivers, thinking about how to become, better and more informed water users and stewards. So lots of ways to get involved. We definitely encourage you to check out our partner pages as well. For 2022, we are really developing lots of ideas about how to spread the word in more physical ways around the city. So what you see here is a photo of part of the Wild Mile. We've got kayakers that are near that Urban Rivers project that's kind of behind the REI over in Lincoln Park. We want to have H2NOW signage up everywhere possible so that people can get the QR code, download it, and understand where, how the water quality is changing in real time. So we've been talking with partners like the Architecture Foundation that does the boat tours, because we'd love to have QR codes on the boat. So if you have an idea for a place where you think we should have H2NOW signage, because it's a place where people are, it's a jumping off point for interacting with the river, please let us know. We'd love to talk about that and think about how we can really get the word out as much as possible. We will have a plan for water sampling volunteers in the 2022 season. This is something we've done in the past. It is a great way to get involved with current. We're out there weekly during the summer season doing those grab samples and helping to make sure that the gauges are still properly correlated and telling us what we need to know. So if you want to get out there and actually help the team do water quality sampling, that's a really exciting way to get involved with us. And we love having, anybody, students, just people with interest in water quality and rivers, come on out. You don't have to have any baseline understanding. We'll explain everything you need to do. And a lot of it is just making sure, you've got clear path to get those samples out of the water. So that's a really fun way to get involved with us. And of course, you can support Current. So we are a nonprofit organization. We are always looking for partners. We are always looking for other sponsors and stewards of this work. Again, H2NOW Chicago is made possible with really flagship support from the Chicago Community Trust. Our BlueEDU work is supported by the Walder Family Foundation. So we are always interested in finding new partners to help us build out and expand from this platform. We also have a really exciting event coming up just right around the corner here in October. Starting October 11th, we'll be kicking off the second Chicago Water Week. And this is something that Current really does in partnership with all of our other partners here in Chicago that care about water. I've already named many of them today. There's more partners signed up to do programming during Water Week, but it's really a week where we put Chicago's water leadership in focus. And it could be from our research universities and national labs, from our counselor partners talking about the amazing innovation partnerships that we have between other countries and Chicago when we think about sharing the best in water technology. H2NOW Chicago is a great example of global water innovation and collaboration, because we have technologies like the Proteus probe, which is a company out of the UK that makes puts all of that together. We've got our Israeli partners, IOSite, that help us with our analytics. So really, water innovation is a truly global endeavor. And so we're always really excited to highlight some of our partners that are not in Chicago, but work with us here to help bring technology into the real world and solve our water challenges here. We have a lot of advocate partners that work with us, others that are interested in monitoring, like the Great Lakes Observing System, our partners at Audubon Great Lakes, and so many others that we know we'll be adding every day here as we head up into Water Week. So the QR code will take you to the event page. If you have an idea for a program, it's not too late to get one on the calendar, but we're really excited again to have another week of mostly virtual events celebrating our water leadership in Chicago. So that is the end of the presentation on H2NOW. If there's no other questions, we'll let everybody get a little bit of an early start on lunch. But just to say again, we're so grateful for all of you and your interest in H2NOW Chicago. Current is powered by partnerships. We can't do this work alone. H2NOW is a great example of that. And we just look forward to seeing you out there, figuring out how to use the river in more informed and engaged ways in the rest of this season and beyond. So thanks so much for coming today. Thank you to the team for all the work bringing this to life, and to all of our partners who make it possible. Thanks so much, everybody.